Our agenda, please be invited again to take the floor for the presentation on seed selection, seed bank, and indigenous seeds. Thank you.
Okay, thank you. Thanks again, Dr. Krishna. I hope the sound is clear, yeah?
It was very clear for almost all the period of your presentation. Just one short interruption was for, I don't know, maybe 1 second or something like that.
Okay,
so
okay.
no problem. I think everybody finally understood from the slides.
Okay, thank you, Dr. Krishna. I'm very happy now. Okay, this is the second presentation from my side. I'll briefly talk about indigenous seed, seed selection, and seed bank. Partly some informations have been already shared by earlier two faculty members, so it will be easy for me as well to explain very briefly. Okay. I'll describe briefly what is on indigenous seed and how we can select this seed, and if we have enough diversity, and how we can conserve for long term this diversity. First, let's talk about the indigenous seed. With these presentations, the limitation of the definition of seed is, it is simply the planting materials which is produced by sexual reproduction. A sexually propagated crop have not been defined as seed in this my presentation. So indigenous seed, we all know, are those seed produced and growing or living naturally in a particular location, and these are selected and managed by local people, and they have very high level interdiversity and heterogeneous, and it is well adapted to the growing area. And based on the nutrition analysis, we found most of the land masses are very nutrition dense. And if you compare the nutrition yield, we are started talking about the two terms, nutrition yield and health index yield. In the past and even now, we always talk about the grain yield, but it is better to talk about the nutrition yield and health index yield, because many people are now more concerned on the nutritional value and how healthy the food we are eating. Therefore, instead of talking about the grain yield, it's better to talk about the nutrition yield and health index yield. We in Nepal also compare the nutrition yield and health index yield with improved variety, so-called modern variety. And we found both nutrition yield and health index yield are higher in the land masses compared to the modern variety. And one issue, we now in the modern agriculture systems, we generally produce seed in different locations, and they imported or exported or transported to other locations to produce. That means the seed production and grain productions are in different zones. That may be some difficulty to adjust the genes. That means the adaptability of such systems may be not fixed, or it may be vulnerable. Therefore, we in Nepal, we have started the systems that where we can produce the seed, and for grain production, the seed from the same locality should be grown, so that the adaptability or the effort to adapt the any area can be minimized. And these indigenous seed are, we all know, the source of the different genes that resulted in the green revolutions, that we have so many different kinds of options in the market now. That's because of the different genes from the local land masses. Therefore, we should say the land masses or indigenous seed are the foundation of agricultural science. We can divide different types of seed based on different criteria. For example, based on breeding perspective, we have indigenous seed, local variety, land disease, high yielding variety, hybrid seed, and genetically modified. I have defined here very briefly. There are some minor differences among these. And based on the conservation perspective, we have orthodox seed and recalcitrant seed. And these are the very useful to know for conservation. For example, if you have orthodox seed, you can decrease the moisture less than 12%, and then you can store for a long period. Long period means more than 100 years. And if you have a recalcitrant seed, you cannot store for long times, or you cannot reduce the moisture content of the seed. You need to plant immediately. And also based on the botanical perspective, we can divide the seed into monocot and dicot. I think you all are familiar with these two types of seed. One of the uses of the landraces is in breeding. This is the one example of called Kumal 4 variety. This is one of the very popular rice variety growing in Nepal. And this variety has been developed using 13 different kinds of landraces. And these landraces were originated from 8 different countries. So richful genes have been incorporated in the Kumal 4. You can see here, I have estimated even the total genic portions. For example, Sina is the landraces that have been used to develop the Kumal 4, and its genic contributions, the total genic contributions of Sina is 7.9% for this Kumal 4, and it is from China. And similarly, I have estimated the genic portion of each landraces that doesn't have further tree or further ancestor. So this is the complete pedigree of Kumal 4, and this is very useful even to convince the farmers, students, or even researchers how it's difficult to get the better variety, because here we have to cross the many times, so many times, using such many different kinds of landraces that were collected from different countries. So how many landraces we need? This is the one example. Therefore, the main uses of indigenous seed is to develop the variety depending on the different criteria or depending on the different sites. We also need to assess the status of agrobiodiversity. The red listing is very common in forest area, and we agriculturists never tried to impose or to apply the principle of red listing before 2000s. And in Nepal, we started red listing steps, and we also red listed. This is very useful to protect or to conserve the land assets. If you define or if you identify the red list, then immediate action is to collect this for long-term conservation. And this is the very simple. We can have a very complex methodology. I have written this, and it is already published. And in previous days, some faculty member already shared, so I'll not explain detail here. And similarly, we can use this we call trait distribution analysis to identify the unique landmasses. And this unique landmasses is also we call endangered, and immediate actions need to collect for long-term security. Another area, because if we see the development activity, many agricultural land are in danger. For example, sorry. For example, many organizations distributed modern variety, and there may be different kinds of heavy disease and some commercializations or monoculturing and migration of farmer, different kinds of land use change policy, and all these factors converted such traditional agriculture land into red zones. We call red zones. Why red zones means the landmasses of this area are now in danger. If you cannot collect these landmasses, in the near future, these landmasses may be eroded. That's why we call red zone. Therefore, we need to define the red zone and need to take the actions for collection or for saving the endangered landmasses. And then how we can conserve. The conserve, either we can conserve through continue growing. If you add the value to the landmasses, farmers wants to grow continue and it will automatically conserve. Second options, if we use in breeding and part of the genes will be conserved. And another one is if landmasses are not competitive, we need to make them competitive. We call landmass enhancement. And if you improve the some deficit traits of landmasses, many farmers wants to grow and it will automatically conserve. And the last one is long-term conservation, we call seed bank. In seed bank, we generally conserve the orthodox seed. The second topics is about seed selections, and the major basis of seed selections is mutic events, mutations, heritability, and genetic variation. Our feature, our working modality should have such facility that could happen these events, mutic events, mutations, and we need the highly heritable traits, and the genetic variation is the must for selections. And if we analyze the mode of reproductions, we have two types, one sexual, another is asexual. In asexual, the selection response may be very low, but in sexual, we have two types of crops. One is autogamy, another one is allogamy. If you impose the selection in autogamy, its output is to have the homogeneous or homozygotes and homozygotes. In case of cross-pollinated crops, our selection target is to have heterogeneous and heterozygous populations. Response to selection is very important to advance the materials, and this is the best distribution of base populations. And based on these base populations, we can estimate the population means and selected portions means, and based on these two means, we can estimate the selection differential. These are the selected populations. And for the next generations, from the selected populations, our distribution pattern will be like that. And we have a means of this selected progeny, and based on this value and this original means, we can estimate the response to selection. Here's a formula, you can estimate easily. And for each different kinds of population, we can estimate this population. And similarly, because many traits are correlated, therefore, handling the single trait might not be resulted in the good genotypes. Therefore, we need the correlated response. That means the change in one trait characters through indirect selection on an associated other characters, for example, X. This is the formula that anyone can estimate easily. And for selection approach, we may have different options. We can select participatorily or non-participatorily. We can have selection exercise either on farm or on station. We may have different kinds of population, landmasses or exotic. My emphasis is to give to the native diversity or native landmasses to improve. You can either use naturally cross populations, or you can make artificial cross. And the selection may be either based on populations or individual selection, or you may have direct or indirect selection. These are our selection exercise in our field. Here you can see so many different kinds of amaranth landmasses. These are the mixture of landmasses and so many diversity and so many options to select. And the beauty of this heterogeneous population is to accelerate the evolution. If you accelerate the evolution, the chance of getting most suitable genotypes for the next season will be happen. Our selection, if we see the development or modern agriculture systems, we more oriented to develop the monomorphic genotype. We call monomorphic. These are the monomorphic genotypes. And if you grow the monomorphic genotypes or homogeneous or monogenotype, there is very less diversity. Diversity is almost zero. If you have a less diversity, there is a more vulnerable to disease and other climate stresses. In contrast, for example, this is the land masses, popular land masses we call Jumli Marsi, that are grown in the highest altitude in the world. We call Jumla. And this is the great source for cold tolerance rice, and more than 50 countries in the world, they are using these genes from this land masses. You can see here so many intra-level diversity. If we have such diversity, then we may have less disease and less vulnerable. Therefore, our target should be the variety that have a more diversity, that can resist different kinds of stresses, and that can have a more adaptability. And that also accelerate the evolution over the year, and farmers can maintain seed themselves. And for selections, we can use 5 units. For example, first you can select the crop field, and then within crop field you can select plot, and then plant, and then flower or spike, and then fruit or seed. Our general habit is to select fruit or seed, but better is to start from the beginning. And similarly, we can select 5 different crop stages: seedling stage, vegetative stage, flowering stage, maturity stage, and storage stage. Our common practice is only maturity stages. Our suggestion is to impose selection in all the 5 stages. We can use different selection tools to enhance or to get the better results. For example, you can use correlation coefficient, path coefficients, heritability, and different kinds of variation component. You can also use stability parameters, diversity index that I taught in the previous lecture. And you can also use check variety, and you can also estimate different kinds of selection indexes and yield component. And similarly, we can accelerate your work analysis using software. These are some software that are very useful to select the genotypes, and nowadays most commonly used by breeder is ggBiplot. And this is not free versions, but you can use this R package. This is the free, and almost all functions are available in this. And there are many packages and software that you can use. Some of them are free. For the farmer's perspective, the farmers wants multiple better trade, but we breeders generally target to single trades. Farmers, for example, wants high yielding without external inputs, early maturity, good cooking, pest resistant, medium height, drought resistant, strong stem, good tillering capacity, erect leaves, large seed size, non-shattering, cost beneficial, fodder value. So many different kinds of trade we have to add in a variety. Therefore, we need the multiple genotypes for covering all these different kinds of trade. And farmers will be very happy if they can get such kinds of material having these 13 different kinds of trade. We can, based on the class, for example, if you have a monogenotype, you have 3 genotypic class, and based on the 3 genotypic class, we may have 3 types of selections. The first one is stabilizing selection. That means individuals with mean equal or close to particular means are selected. These portions is selected, and it is called stabilizing selection, and their population would be like that, the next progeny population. And the second we call directional selection. That means selection either in the directions. of higher mean or lower mean. So you have a lower mean here and higher mean here. You can either use this higher mean or lower mean. That means direction selection. And for the next portion, if you select the higher portion, then you will have such kinds of distribution. And the third one is called disruptive selection. That means selection of more than one classes. You can select this class as well as this class and combine, you will get the two mode-like distributions. For selection method, there are many methods available in the Google and in the textbook. Most commonly used is mass selection. This is very simple one. Farmers, breeder, students, they can do. And this, it has a very beauty. It has heterogeneous. It developed the heterogeneous population. And we also have another called pure line selection. Pure line selection is also very simple, but it develop completely homozygote that may be risky in the future or even in the new environment. They cannot evolve the new allele or new genotype, through this pure line selection. Another, the new concept that we are using in Nepal, we call class bulking selection. This is very simple one, and it combines the different kinds of classes together in a single genotype, single population. And this helps to accelerate the cross-pollinations, ultimately accelerating the evolution of genotype. Different kinds of genotype. There may be best ones or there may be worst one. And we always select the best one and combine them together. And it keeps evolving and it keeps increasing the performance for a particular trait. If possible, I suggest to estimate the mixing ability. We can estimate the mixing ability similar to general combinability and specific combinability, like in genetics. And based on this mixing ability, we can combine. The last section is about seed bank. I will not take more longer. It's already talked. The seed bank, simply the place where orthodox seeds are stored to preserve genetic diversity for the future as well as present, along with information. These are some examples of seed banks. And some features of general banks is banking of many different kinds of seed, and the germplasma as well as information is access to all for long-term security and able to trace backward and forward. Any germplasm movement we can trace backward either as a forward. We can divide the seed bank in different categories based on the different criteria, and seed bank is the part of the gene bank. And the first one we can say on-farm seed banks. And in this, the seed generally interact partially with the nature because some months the seed are stored in the house and they could not interact during that period. Therefore, they partially interact with the nature. And for example, community seed bank, household seed bank, village level seed bank, these are the on-farm seed bank. And these are generally for short-term storage. And another we call ex situ seed bank. And here almost the seed never interact with the nature. This is static, and there are global seed bank and national seed bank. And global seed bank is generally for long-term storage. And national seed bank, they may have different options. For example, for long-term storage, medium-term storage, and short-term storage. And the third one is in situ seed bank. That means completely the seed in situ bank interact with the nature. Even in the dormant period, they live within the soil. That means they always keep interacting with the nature. Therefore, we call in situ seed bank. For example, natural seed bank, national park, and agro-gene sanctuary. We have also started the agro-gene sanctuary. That means we never reintroduce the domesticated crops in the forest. And we have started reintroducing the domesticated crops into certain area in the forest. And we always let them interact with the nature. And we are observing what will happen in the genetic makeup. And for storage condition, in the community seed bank, they usually store in the normal room temperature, like that. This is all local materials to store. And in seed banks, we usually store in -20°C using airtight condition, and this is for very long duration. And in the natural seed banks, this is completely natural conditions. They always interact with the nature, and they try to evolve better genotypes after interacting with the nature. This is the workflow of the activity in the seed bank. I have just listed here. You can read later. And in the gene banks, our major activity is regeneration and distribution. And regeneration, during regenerations, we always consider not to lose the diversity. Therefore, we have to maintain the good size of plant population for regeneration. And these are the germ plasma flow steps in the gene bank. And distribution is the major work in the gene bank. And we generally made accessible to all, to any times, but only small amounts is available. These are the database. You already saw some of these: GeneSeed, Green Global, NIAS Gene Bank, Salboard, UNESCO, and you can source different kinds of information, including characterization, evaluation data, DNA data, and some image as well in this website. And you can also ask seed for research. These are some published papers that I already shared, and you can read this one for further details. And these are some further readings and some useful web links for enhancing your knowledge regarding with this indigenous seed and seed selection and seed bank. And these are some very useful genotypes, useful software that are very useful for selecting the genotypes after generating data. You can use this, and it will be very helpful to identify the selection indexes. So that's my all presentation, and I like to all the participants, audience, for your patience, and Dr. Hasrat and the Grassroots Institute and all partners. And let's make native landmasses competitive. This is my statement, and not to try always to depend on the exotic landmasses. The nature had gifted so many landmasses for a particular site, and these landmasses are well enough to fit this particular site. Only the problem is to make them competitive. Thank you all, and Dr. Christina.
Thank you, Dr. Joshi, for sharing with us this information regarding the selection approaches of seeds, and well, by making us having a higher awareness about taking care on these choices, like keeping in mind that genetic uniformity leads to genetic vulnerability, and that we have to ensure genetic increasing diversity, so that as well as to share with us the, let's say, lucky 13 criteria for farmers when choosing the seeds. Two of them are high yielding, of course, and but as well as good taste when cooking are two of these lucky 13 criteria. And also a personal thank for, if I observed correctly in one of the slides in your presentation, it was the Great Everest just nearby. And well, I salute you as somebody living so close to this very famous mountain. So thank you very much for the information, and congratulations for the work that you are doing in your institution.
Thank you so much, Dr. Krishna. Bye.
Now, if I understand correctly, Professor